Hello everyone, my name is Chenyi Li. I'm pursuing my PhD under the supervision of Dr. Yajun Yan at the University of Georgia. Today, I want to share our research work with you. As you may know, microalgae hosts can natively generate a great precursor chemical pool and cofactors from renewable feedstocks, offering a microalgae-based platform for production of a variety of natural or non-natural chemicals. Extending from existing microalgae biochemical infrastructures, carbon flux can be streamed into products of interest via purpose-driven biosynthetic pathways. Amino acids are constitutional units for proteins and also one of the major endogenous metabolites in almost all microorganisms. However, their utilities as building blocks for chemical productions have been overlooked. Protein hydrolysis from protein-rich biomass or protein waste from food processing industry and industrial fermentation residues serves as inexpensive and readily available external sources for amino acids. Therefore, the structural diversity is of achieving high titers in microorganisms and ever-increasing accessibility from protein feedstocks have substantiated amino acids with potential as versatile starting scaffolds for various industrially relevant natural or synthetic products. In recent decades, intensive efforts have been taken towards converting branched-chain amino acids to advanced alcohols or acid, and aromatic amino acids to medically or nutritionally important chemicals like flavonoids or alkaloids. However, the development of efficient biocatalytic routes to produce functionalized chemicals from charged amino acids, which account for over 40% of all amino acids in protein hydrolysis and over 80% of all amino acids market, are comparatively less tapped. Charged amino acids are bifunctional terminal groups, making them recalcitrant to be directly metabolized through central carbon metabolism. Additionally, the paucity of natural or synthetic pathways to utilize charged amino acids significantly limited the valorization. Here, we describe the development of a metabolic platform that enables direct conversion of charged amino acids into C3 to C5 diols. The proposed AA to diol platform proceeds through two parts. The upstream degradation of charged amino acids into corresponding C-1 omega hydroxylic acid, followed by the downstream reduction of omega hydroxylic acids into counterpart diols. Typical charged amino acids, including aspartate, glutamate, and lysine, share a common degradation process of sequential decarboxylation and transamination to counterpart C-1 semialdehyde, which are further reduced to omega HAs via aldehyde reductase. With the existence of well-elucidated native or heterologous AA catabolism supporting our hypothesized platform, the omega HA to diol conversion becomes the most pivotal step for diol conversion. Thus, first, we need to validate the feasibility of this pathway by testing the last step, which is a conversion of omega HAs to corresponding diols via reduction catalyzed by carboxylic acid reductase and aldehyde reductase YKHD. The classic pathway of omega HAs to diol conversion harnesses a CoA-dependent reduction route that involves activation of carboxylate group by a CoA transferase, followed by reduction of the CoA intermediate to aldehyde and then diol by a bifunctional alcohol dehydrogenase or CoA-dependent aldehyde or alcohol dehydrogenase with the input of 2-NADPH. We here propose a CoA-independent reduction route that utilizes a promiscuous carboxylic acid reductase or CAR and endogenous aldehyde reductase to reduce carboxylate group to alcohol group at the cost of 1 ATP and 2 NADPH. To experimentally assess the pathway performance, the CAR-based reduction route was expressed in E. coli and tested by feeding C3 to C5 omega HAs. When fed with 3-hydroxypropionate or 3-HP, the recombinant E. coli strain harboring the PCAR plasmid produced 0.97 gram per liter 1,3-PDO from 3 gram per liter 3-HP in 36 hours. Despite the relatively low conversion rate of 3-HP, the CAR showed high conversion capability toward 4-HP and 5-HV, which produced 2.6 gram per liter 1,4-butanediol in 24 hours and 2.7 gram per liter 1,5-pentadiol in 12 hours, accounting for complete conversion of both substrates. The bioconversion results underline the high performance of the CAR-based pathway in omega HA to diol reduction. Noteworthy, CAR exhibits a catalytic preference towards longer-chain omega HAs, which is consistent with its preference toward long-chain fatty acids as reported before. This was further confirmed by the increase of the specificity enzyme activity of CAR towards substrates with increased chain length, as you can see from the enzyme assay results. With the validation of an efficient omega-HA to Diol conversion pathway, our second goal is to assemble the upstream AA degradation pathways capable of converting charged amino acids into C-1 omega-HAs. For aspartic acid degradation, a synthetic 3-HP pathway was constructed consisting of L-aspartate alpha decarboxylase (ND), beta-aniline pyruvate transaminase (BAPAT), and malonate semialdehyde reductase. The native glutamate metabolism, also known as gamma-aminobutyrate or GABA pathway, generates GABA via glutamate decarboxylase GADB, and GABA can be transaminated to succinate semialdehyde by gamma-aminobutyrate aminotransferase GABT, followed by reduction to 4-HB via endogenous NADPH-dependent ADH like YQHD. As for lysine degradation, a heterologous three-step lysine degradation pathway from Pseudomonas butida, known as AMV pathway, was used. It converts lysine to glutamate semialdehyde with lysine 2-monooxygenase DAVB and 5-aminovalerimidase DAVA. Further reduction of the glutamate semialdehyde by endogenous ADH will release 5-HV. When fed with 3 grams per liter corresponding amino acids, all 3 strands show successful production of corresponding hydroxylic acids. Validation of both upstream and downstream parts made it possible to achieve the direct conversion of charged amino acids including aspartic acid, glutamate, and lysine to Diols. Therefore, we combine these 2 pathways and tested the direct conversion of amino acids to corresponding Diols. The E. coli harboring the 1,3-PDO pathway, or strain 3-PDO1, did not show any 1,3-PDO production if no exogenous ASP was fed, while it produced 276 mg per liter 1,3-PDO in 48 hours when fed with 3 grams per liter ASP. This validates the functional expression, a low activity of the complete 1,3-PDO pathway, likely because of the cumulative efficiency of the upper 3-HP pathway and the low activity of the CAR towards 3-HP. Upon expression of the 1,4-PDO and 1,5-PDO pathway in E. coli, the strain produced 588 mg per liter 1,4-PDO and 123 mg 1,5-PDO without glutamate or lysine feeding. While feeding 3 grams per liter glutamate only resulted in a slight increase of 1,4-PDO titer, feeding 3 grams per liter of lysine into the strain dramatically increased the titer of 1,5-PDO by 10-fold, which indicates that the lysine availability is a major bottleneck for the 1,5-PDO synthesis. On construction and validation of the C3-C5 Diol production platform, we sought to broaden its substrate scope by rerouting all potential amino acids into counterpart Diols. E. coli natively harbors amino acid degradation network that enables conversion of 4 additional amino acids into our platform. All of these amino acids, hereafter referred as Diol-convertible amino acids, have previously remained unexplored for deriving new chemical entities due to the structure complexity. We then adopted this native amino acid degradation pathway, as shown on the slide, to enable Diol production from Diol-convertible amino acids. To this end, we co-transferred each Diol pathway plasmid and AA degradation pathway plasmid into E. coli, and the resulting strands were cultivated and supplemented with 3 grams per liter amino acid substrates for 48 hours. When fed with asparagine, the strain produced around 145 mg 1,3-PDO. Regarding 1,4-PDO conversion, the strain with 1,4-PDO pathway and the corresponding amino acid degradation pathways enabled a titer of 650 mg from glutamine, 903 mg from proline, and 648 mg per liter from arginine. The bioconversion experiments demonstrated that with overexpression of AA degradation pathways, more amino acids, including asparagine, glutamine, proline, and arginine, could be converted to counterpart Diols. Since aspartic acid is a direct precursor for lysine biosynthetic pathway, we also tested if feeding aspartic acid could lead to 1,5-PDO production. Unexpectedly, feeding aspartic acid to strain could just slightly increase the 1,5-PDO titer. Combined with the lysine feeding results, this further suggested that the ASP to lysine pathway is a limiting step for the synthesis of 1,5-PDO. The establishment of amino acids to Diol platform expands the feedstocks to amino acid and also implies the feasibility of direct Diol production from sugar by connecting it to the endogenous amino acid pathway in microbial chases. To test that, we first choose to demonstrate and optimize the 1,4-PDO production from glucose in E. coli. The C4-Diol 1,4-PDO is a large volume commodity chemicals with extensive applications in polymer industry and its bio-based production at the commercial scale have been achieved via the CoA-dependent pathway with extensive enzyme screening, host engineering, and fermentative process optimization. To increase the 1,4-BDO production, we sought to improve glutamate decarboxylation efficiency. The wild-type glutamate decarboxylase GADB of E. coli exhibited catalytic activity in acidic conditions, and its mutant GADB star E89Q and DIRT 452 and 466 showed improved activity at a neutral pH. The glutamate catabolism requires PLP as a cofactor. To increase the PLP supply, we overexpress a short ribose 5-phosphate-dependent PLP pathway from Bacillus subtilis that involves two enzymes, the PDXS and PDXT. An additional benefit from the PDXST pathway is the conversion of glutamate from glutamine, which can be taken up by 1,4-BDO pathway. We also considered increasing carbon flux to glutamate by redirecting carbon flux from glycolysis to TC cycle. We overexpress an NADPH-insensitive citrate senses mutant, the GLTA star, and the phosphor and pyruvate carboxylase (PPC). When cultivated strain 4-BDO1 in the M91 medium, only 588 mg 1,4-BDO can be observed. The introduction of the PDX-ST resulted in a 14 increase, with the titer of the strain 4-BDO2 reaching 675 mg per liter. The carbon flux redirection by GLTA and PPC increased the 1,4-BDO titer by 45.6%, that of the control strain 4-BDO1, confirming that the redirecting carbon flux to glutamate could augment glutamate availability and thus increase the 1,4-BDO production. When further introducing the PDX-ST into the strain 4-BDO3, the resultant strain 4-BDO4 afforded a 1.41 gram per liter 1,4-BDO titer in 48 hours, representing a 2.4-fold increase of the titer in relative to its parental strain 4-BDO1. As a proof of concept demonstration, our results highlight the potential of reducing 1,4-BDO from glucose via the glutamate degradation machinery in microalveolar host. As a safer replacement for 1,4-BDO, 1,5-BDO is also a high-value and non-natural short-chain diol with wide applications as monomer for fibers and polyescenes. Currently, 1,5-BDO is chemically produced, while direct biocatalytic routes from renewable carbon source to 1,5-BDO have not been reported. With a lysine conversion platform, it is possible to fill the gap by achieving de novo production of 1,5-BDO from glucose. The insufficient lysine supply is a major rate-limiting step for de novo production of 1,5-BDO, as observed earlier. To enhance the downstream lysine pathway, we introduced the LYSC and DAPA on the medium copy plasmid combined with the lysine degradation pathway. Meanwhile, to desensitize the lysine-mediated feedback inhibition, the lysine mutant T253R and the DAPA mutant H56K or E84T were also recruited, yielding strain 5-BDO3 and 5-BDO4. The strain 5-BDO2 expressing the wild-type lysine and DAPA produced a titer of 322 mg per liter to 1,5-BDO, representing a 2.6-fold increase to that of the parental strain 1,5-BDO1. Whereas strain 5-BDO3 and 5-BDO4 could not further increase the 1,5-BDO production in comparison with strain 5-BDO2, with titers of 2.15 and 311 mg per liter respectively. This suggested that the lysine degradation via the 1,5-BDO pathway might alleviate the lysine-mediated feedback inhibition, which in turn leads to higher 1,5-BDO titer with the wild-type pathway enzymes. Therefore, to reinforce the lysine degradation, we incorporated this pathway into a high-copy plasmid. Notably, feeding 3 gram per liter lysine to the strain containing the PCAR and the P5H5 plasmid enabled the production of 1.67 gram per liter 1,5-BDO, reaching a conversion efficiency of 72%. When co-transferring all these three plasmids into E. coli, the resultant strain 5-BDO6 significantly increased the 1,5-BDO titer to 0.86 gram per liter, representing a 7-fold increase than that of the starter strain 5-BDO1. Finally, to increase the supply of lysine-separate precursor OAA, we knocked out the ICLR, which encodes a repressor regulator of the glyoxylate shunt that serves as an alternative mechanism to supplement the OAA. The final 1,5-BDO titer produced by strain 5-BDO7 with ICLR deletion reached around 0.97 gram per liter at 48 hours. And interestingly, this strain also accumulated the least acetate in the culture. Taken together, our results established and optimized the 1,5-PDO pathway in E. coli and demonstrates the first de novo production of 1,5-PDO in microbial host. To summary, in this work, we successfully established a CoA-independent pathway to convert omega hydroxylic acid to short-chain diols by harnessing promiscuous activity of carboxylic acid reductase. Then, using the CoA-based pathway, we established a platform for converting 3 charged amino acids to counterpart diols. We also demonstrate the general applicability of the platform by converting a total of 7 amino acids to counterpart C3 to C5 diols. Finally, as proof of concept, we demonstrate the industrial potential of this platform by engineering de novo biosynthesis of 1,4-PDO and 1,5-PDO. I'd like to thank my advisor, Dr. Yan, and my lab mates for their help. We also thank the support from the College of Engineering in the University of Georgia. Thank you, everyone, for your attention. That's all for my presentation.